Hello, everyone. Thank you so much for joining. Today, we are here for a very good talk. Dr. Sebastian Raja is going to give it to us. Just a little bit about his excellent background. Dr. Raja is an accomplished biophysicist and microscopy expert with over 15 years of experience across academia, research, imaging technology. He specializes in high resolution fluorescence imaging to study cellular and molecular mechanisms and holds a Ph.D. in biophysics and cell biology from Universidad Autónoma de Madrid. Dr. Raja has worked internationally in Finland, Israel, and India and currently leads applications at Leica Microsystems, which focuses on advanced microscopies and user training. He has published extensively, won awards for his application expertise, and is recognized for research on HIV-1 nuclear trafficking and imaging technology. Dr. Raja is also an active educator, regularly teaching advanced microscopy at top Indian institution. Uh, I can keep going and going, but here we have Dr. Raja. Thank you so much for doing this. And You can start, and we'll have a talk first. And please, everyone, if you have questions, put it in the chat box and we'll address them at the end of the meeting. All right, Dr. Raja.
Thank you, Deepak. Thank you so much. First of all, I have to thank to the organization, and it's a big honor to me. And thanks, first of all, for this great opportunity to discuss with the physicians. In fact, I'm quite far from the clinical side, but again, I'm working with clinical samples and everything. Not exactly with any of the clinicians directly. I hope the talk will be enjoyed because it's going to be the very advanced biological and quantitative cell biological applications for image processing and analysis by using AI-based system. So at this moment, I'm just closing the video due to the speed of the net connection. Don't mistake me. So I can share the screen. Can you let me know the screen is
Yes,
you can get
it's coming.
Okay, great.
It's okay. Now we can see it.
So, okay, fine, great. So these are some of the images from our it's Leica microsystem. So it's before going to the talk, I can give you a very short intro about the Leica, because Leica is a German-based microscopy company, and we have the competitor with the Zeiss as well. So, and we are 176-year-old company basically. So for the past 175 years, we are working in a imaging area, and to like most of the around more than 60% of market share, more 50 to 60% of market share in the microscopy, especially on life sciences. We are so almost a lot of research articles and everything. The images is coming from his Leica. So we are so proud to tell that. So before going to this, regular imaging strategy, if you go to the strategies of any kind of imaging, so you have sample preparation, microscopic acquisition, and image analysis. So in the three parts, we already contributed. Of course, the sample preparation and image analysis will be highly dominated for any scientist to publish a paper or publish an article in a very high impact journal for cell biology and everything. And of course, the microscopy acquisition, it's very important by the way of to capture the images in the proper or the correct scientific way. So today we are going to discuss more on imaging analysis. So you can see here, there are different multimodal imagings are available in cell biology. So like almost you are mainly working on PET and MRA and ultrasound and optical imaging and everywhere. But today we are going to talk about the super resolution and the confocal and other super resolution techniques. It's not an electron microscope, but we can call it as a nanoscopy basically. So we can go through this kind of techniques because it's going to be the very high resolution with the spatial as well as the temporal. If you go deeper and deeper, so we can go ahead. So we are not going to talk about any organism level, but at the same time, we are going to talk about the molecular level and the organelle level, cellular organelle level as well. So like the first question is everyone has that, like, what is your sample? So general biology, people can use in bacteria or otherwise the mammalian cells. It may be the tissue, it may be the protein or any kind of biological sample. So people can segregate as different types of cells. Of course, if you talk about the cells, there are different organelles are there, inside there are various organelles. But the question is how the organelle, is it structurally resolved? So what kind of information inside on every organelle, how it is separated, and how it is distributed, like what is the concentration. If it's in the live condition, is it that it is aggregated, or it is interacted with other molecule or not? So these kind of n number of questions were developed by molecular biologists or genetics or biochemists, or like anyone can go and study inside into the cell to helpful for the humanity. So that's the reason we were starting in the sample level. So the basic idea in microscopy, it's like cutting the apple. So you can see that we are cutting the apple by the knife, but here we are cutting the apple by optical knife. So we call that Z stacking or the three-dimensional imaging. You can call that. It's called as optical sectioning basically. So I can show you my laser pointer. Hope you can understand it. So once you cut it from the top to bottom, yes, you can have a lot of slices. Once you join together, you will get the three-dimensional image. That's what the real fundamental idea of taking the images in a very high resolution, the three-dimensional space. So now you can see here, there are different types of microscopy techniques are available. People use total internal reflection to study only the surface receptors and everything. Or people use a wide field fluorescence microscopy to illuminate throughout the cells. And they can go in a very fast imaging by using a camera. And people use the laser scanning confocal microscopy by imaging by using the point light source for the very high resolution. And nowadays, people use some kind of nonlinear optical microscopy like multiphoton techniques and everything, going very deeper and deeper in the tissue. They can go up to 1 mm in a live tissue and try to image. So these applications are quietly used in neuro-based neurosciences as well as in cancer biology or developmental biology. Everywhere, widely people are using again and again. So now, once the data are generated by using these images, so how we can analyze these images? Like what information we can extract? That's the interesting point here. So now you can see these images here. Just, I'm sorry. There are two different images you can see here. One is with the regular confocal microscope image that's called as conventional photon counting image. And this the Stellaris is our confocal microscopy named as things. Now you can see there are very fine details here inside that. There are a lot of very fine new particles or the new protein aggregates are trying to go here and there. This is a live image data. But the image looks very complicated because there are a lot of spots. Every spot has some movements. Every spot due to the time scale is structurally changing inside. So like how to elucidate all this information. So like what kind of microscopy we use is the second part. But once you got the image from that, how you are going to analyze. So I'm not concentrating on how to capture the data or how to prepare the samples, not. So I'm only going to concentrate on how to analyze this data. So what kind of information we can get. So every one of them are interested by using high resolution imaging. So once you can see there are two proteins are together functionally, is it possible to resolve the two proteins together inside the mitochondria, inside any organelle? That's one of the very fascinating in the advanced biology now, cell biology. It's called as cellular structural biology or cellular structural biochemistry. You can call whatever the name you want. So the aim resolution says that in any one of the conventional microscope has the XY resolution of only 200 nanometer. That is called the diffraction barrier of the microscope. So once you are killing or once you are breaking this diffraction barrier, that's called as super resolution microscopes. So people are trying to do the super resolution because 2015 Nobel Prize in Chemistry is dedicated for super resolution of the microscopes. There are different techniques are available in the world to go the resolution up to 1 nanometer in live cell itself. That is big revolution in the world basically. So like seeing the molecule at the live state for the very high resolution, it's not an easy task. So that's one of the very fundamental question everyone will ask. Everyone is enthusiastic to understand the basics of the biology, what's really happened. So now you can see this is the point split function or the resolution limit I would have tell. I don't want to go more physics, but people are starting in the wide field like the bigger one. Once at this moment in the single location, they can go in a very point light source. I would have called this area is around femtoliters. That is, femtoliters means like zero. After zero, you can put like 15 zeros. Then you put 1. So 10 to the power of minus 15 liters, small volume area of people are illuminating and trying to get the information and generating the structural images. That's very fascinating, very advanced cellular structural information, what we are gathering or collecting from the confocal microscopes or any other super resolution microscope like PALMS, CHOMS, STED. So we have STED microscopes and everything, so we can do that. So this is one of the example what we are doing here in the regular confocal microscopy. We are trying to do the adaptive deconvolution protocol. By using the adaptive deconvolution, we are achieving the very high resolution images. So now you can see this image, for example. So with the raw data, you can see it looks like more background. Here you have the much background. We are not able to resolve anything. But with our lightening protocol, it's called as adaptive deconvolution super resolution approach. Yes, we are capable to resolve, or otherwise we are capable to see all the small dots here. You can see which looks very clear. For example, now once you want to resolve this, we have to do the deconvolution. You can see the example here with the regular confocal data. This is the actin inside cellular, like cytoskeletal image. Maybe you have seen before also, but it looks very excited to see. You can see it is like a bones for the cells. Basically, it's a cytoskeleton protein. So you can see the resolution between these two. You can see a very fine defined resolution here. So we can go up to 120 nanometer in XY plane. I told that it's only 200 the diffraction, but now in this image we can go up to 120 nanometer. Now you can see this data. It's this is the data from Germany actually. So like you can see with the brain samples, basically the people are trying to see by using the lightening data, they can try to find the spines, they can count the spines. But with the regular microscopy, they can't count because this is the deconvolved data. That's what the super resolution will help to you to understand how the images are there. Now the question is, you can see with other example of the live cell data with the tubulin or with histone protein and with the GPI, you can see in the live, you can see the cells are wobbling here. So every molecule aggregates, you can see inside the cell. So everything is tagged by the fluorescence dye. So after the fluorescence dye, once it's illuminated, yes, we are getting this. So this is the data we got from the HeLa keto cells, and it is published already. Just for the example, I just show this. You can see this. This is the another example with the different organelles like mitochondria, actin, microtubule, and vesicles. You can see inside. This is very fascinating to see the live cell biology. Inside, everything is wobbling and everything is moving. Now the question is it that how much time it takes? If I'm doing any gene knockout or CRISPR-Cas or any drug interaction, what kind of alterations we can see inside? How it is affecting the cellular pathway? So these are the common biological questions are raised by the scientists, and we are trying to address through imaging. Of course, people do the different kind of techniques like mass spec or any kind of gel or any kind of techniques people use, but we are trying to address by using through the imaging here. Now you can see here with this. This is another example with the lysosomes, and we use with the different types of dyes we use. You can see that lysosomes is transporting from one place to other place, and everything is a different velocity. So how you can measure inside this? So that's the very interesting point here with that. So now you can see this is one of the stitched image. We got it, so it looks very interesting. Believe me, this image is a stitched image. Means we take image of every square and stitch together. So that's called as tile imaging or the image stitching. But you can't see that it's a stitched image. It looks like a single image. It's a very high resolved image of one organism we found in our BU. They shared the image, so it looks very interesting. This is simply autofluorescence. It is a natural fluorescence. We didn't add any dyes there. So it's a simple fluorescence. We added and we took this. So now you can see with some kind of live cell imaging. Why I'm taking this introduction? Some of the data what we are going to analyze based on this actually. So this is a simple HeLa cells actually. So this and the C. elegans embryos are here, and the people are trying to overexpress a protein here and try to watch the protein through the time. So now how the expression is changing, how much time the cell is viable, is it the cell's viability, especially the motion, is affecting or not? So these are n number of questions, n number of information you can get from the live cell microscopy. So now this is, that this is a regular cell cycle pathway. You can see that how two cells are doubling with the meiosis and everything. So you can see really well here. So this is from RCB lab. And nowadays people are working in organoids. So people image the organoid, but organoid is a big data analysis. You can see the organoid, but you want to analyze the organoid. That is the $1 billion question. It's very difficult. So now how we are going to analyze this? So that we will see. So these are some of the examples of different organoids from patients also. From patient cells, they take the primary cells and they can build the organoid and they use that as a humanoid basically. So instead of to inject to any of the animals, people try to treat the drugs by using microfluidics or anything throughout like long times and try to find the excretions and the expressions or these things people are trying to do regularly. So now I'm introducing the AI-based, the artificial intelligent visualization of image analysis software, what we are regularly using in Leica. Okay, so very recent advanced software we used for multiple applications. So the IVIA is basically called artificial intelligence based visualization of image analysis. That's the idea of the whole IVIA. IVIA stands for basically. So you can see we can start from the visualization. So the company DR Vision started and IVIA was handled, and now we procure and we are working and integrated with Leica, and we are working extensively for various applications. So like different kind of application we do. So usually it is started from different versions. Now we have an 15 version released, and every version has the new upcomings of various applications. So we do these things. So now, like what IVIA can do here. So now you can see. So people use the 2D, 2-dimensional, 3-dimensional, 5-dimensional rendering also you can do, and you can handle the data for more than 3 terabyte. The 3 terabyte data means it's a very huge data. Still, you want to analyze? Yes, you can analyze it. And third one, this is a cloud-based analysis software because of very long algorithms and everything. So we have our cloud in Australia. So everything is connected to the cloud. So we posted the data here immediately with the help of the cloud. We will analyze it. So software will run in the cloud only. And you can go for co-localization, neuron analysis, tracking, segmentation. Segmentation in the sense, the very deep segmentation will go. I'll explain about the segmentation, how it is. And we have the machine learning and the deep learning approach. Yes, we can do with the very deeper machine learning and the deep learning approach we will do in this. So we can see all these parameters, how we started. So as I already told you, the microscopy you want to analyze, yes, you have to cultivate your sample and image request, image processing, and you have to segment, you have to render, you have to classify, and you can generate and post your data. So that is a very big challenge in the downstream image analysis. Not only just to take the image, just to prepare the sample, but you want to analyze the data to understand what's happened really in the biology. That's really fascinating in the science. So now we can see the first idea is anyone can access this, any kind of image of TIFF format or anything. For example, the PET data, as you say, or otherwise any tissue or any kind of scorings. The pathologists usually do the scorings. So scoring data, everything you can, any kind of images in any format, JPEG or TIFF, you can analyze. That's one of the first point I want to highlight here. And second is you can do the very detailed segmentation. Segmentation in the sense, like for example, if you are incubating a cell with any kind of X drug, so after the drug interaction, it creates some kind of aggregates inside the cell, or any kind of cell shrinkage or organelle changes or dividing, anything happened, you can segment it separately, everything. So that is the very detailed segmentation what we are doing. And then we can go for the plotting and everything in the same platform itself. That's the another idea. So now you can see here, just one of the example I start here now. You can see this example. Sorry. What? Yes. Now you can see this. Oh, I'm sorry. 1 minute. Yes. You can see the brain image. The brain imaging, brain is a very difficult task to understand. You can see inside the brain, there are n number of neurons are there. So we do the 3-dimensional imaging of the brain and trying to analyze this, every individual neurons, and trying to understand how these neurons has any alterations or anything, and we will plot it. This is one of the example I'm showing you. So this is the image analysis we are trying to segment and do. And this is for the virtual reality of the PET imaging. You know that this kind of organelle based imaging, by using the VR apparatus, like virtual reality apparatus, we can found this mouse, and inside to the mouse you go, you can see that there are the smart segmentation of the tumor. We do that. Now you can see in this, we selected this specific area. It's called as pixel classification. Once you selected the area, then you apply that. There is a background. There is a specific tumor formed. So we just selected it. Then we can process the data. Now you can see that after we teaching the IVR, now you can see, you can find easily where the tumors are. Immediately, this will capture. This is based on AI only. So AI will be selecting the based on the intensity or the morphology, whatever you can select accordingly. So they found the tumor area and they target the specific tumor there and trying to quantify the tumor. So you can see that they will do the segmentation in the 3D space. This is a 3-dimensional space in the image. They segment only the tumor area. So from this tumor area, of course, you will get the volume and all the other parameters. Then they can quantify it. So the similar way you can see here, this is the 3D object rendering in the developmental biology. People in the, because this is in the Drosophila, so you can see the space of the zebrafish or the Drosophila, and you can watch this. So there is a variation between the two different velocities. Yes, you can form. You can see this. This is the another one with the cellular analysis. You can segment it properly. You can see there are different types of segmentation. I'm sorry. There are different types of segmentations are there here. What happened? I'm sorry. Sorry. I think something else. Can you hear me? I'm sorry.
Yes, we can hear you.
Just a minute. So I'm sharing. Yes. Something happened. Okay. I think I gave. No. Sorry. Yes. So now you can see here. This is for the single organism. This is the whole organism imaging in the light sheet technology. By using light sheet microscopy, we imaged, and we can see the individual nucleus, individual cells on the top of the surface, and we can quantify this very individual. And in different area, the nuclear morphology is different due to the developmental biology. And this is the another example of the brain. The whole brain in the multiphoton imaging. People went up to 1 mm inside into the brain and tried to image. They can segregate in a different layer, hippocampus, and you can see the dendrite and granules, and you can see the cortical neurons. They can separate and they can quantify that individually. So these are very, very interesting part. What usually we do that. And this is one of the example of the nuclear pore complex on the surface. So these kind of imaging we are regularly doing here in India. People do the nuclear trafficking of any virus or any particle, anything based on the nuclear pore complex. You can quantify the nuclear pore complex here and you can elucidate it. So this is one of the example I want to show you how they are quantifying by using this. So like what kind of quantification we'll do again. So they are trying to segment it within the background and everything, and they can use this data. So this is one of the another imaging process. Yes, people are trying to do. And here you can see this is one of the another imaging thing for the deep learning things, and you can do the denoising. Denoising means a lot of background will be there. That background will be subtracted. So we have the proper algorithm for that to subtract it, and we can segment it appropriately. Then you can go for the analysis. So that's one of the very interesting part we found it. And here you can see this is one of the another interesting is the electron microscope data. So electron microscope data images will get only the non-coloring, like this is like this kind of only the contrast image you will get. But with that we can build and we can segment different organelles inside to the tissue or anything. This is one of the TEM data. Just I'm showing you. It was literally complicated before. Now with electron microscope data, it looks really amazing. So we can segment it properly and do the 3D rendering and segmentation results. We will do that. So that's a very interesting part. What I want to what I want to show you here. And now these are the basic different types of applications. Yes, we are doing it. But now you know about the spatial biology insights. So nowadays people are trying to find there are more than 15 different types of protein or 20 or 40 different types of genes they are trying to target. Yes, we can analyze the spatial biological samples also by using confocal level. People use in the regular wide field level, but in confocal level, we are the one who is in the market can do that extensively. This is one of the example we do with the different types of genes we analyzed, and these genes are all plotted in the same software itself. It's not like that you have to go outside and analyze this. Once you analyze, you will get it. I'll show some of the example here, then you will get an idea. See, now these are the different types of rendering protocols what we used it, and we can do the detection in the cells. You can see here we are doing the very deeper detections here, and we segmented this complicated cells and trying to quantify that. So that's the very interesting part, and we can apply with the different types of presentation of this data, and with the confidence maps. Yes, we are proving like what has internally happened in the molecular mechanisms happen into this. So now you can see here. So this is one of the example of the 3D rendering. So we can do this kind of rendering and trying to find in the three-dimensional space how these molecules are fitted or how it is arranged. Yes, we can do by using our software. And now you can see here, these are some of the examples. For example, the cell nuclear, you want to analyze only the nucleus? Yes. Or the phase tracking? Yes, you can do only the phase of the cell. And the colony analyzer or any kind of wound healing assays, calcium oscillations, or any kind of neuron based. So there are different types of analysis strategies you can do by using advanced AI based software. Because before it was very difficult to analyze a single image. Now you can analyze and apply for a batches also. So people are applying with the very raster image analysis, very advanced and very speed. And you want to integrate any Python algorithm also, you can integrate it and analyze it. So these are some of the examples I want to show you. So how the artificial intelligence is work. Of course, we have to teach to the machine to do like what kind of things we want to do by AI basically. So we have to feed it already. So once you feed, then the machine will learn it. Okay, these are the parameters you want to see. You want to check the size. Then after that, it will apply to different types of deep learning algorithms to find the morphologies and states compared with the standard data. So this everything happened within a second. So like you want to analyze a big organoid sample or any kind of cell biological, any very big large imaging samples, yes, you can analyze it. So that's the very important one I want to show you here. See here now. So you can extract the object characteristics and go beyond the intensity and the size. That you can do. And you can, examples are provided instead of the numerical measurements. Any kind of numerical measurements are analyzed, yes, you will do. And you can characterize by determined by any kind of algorithms. So any algorithms you use and try to quantify it. So now you can see here. So we can classify these objects by using the pixel classifier. Then we can do object classification. So once you classify that, we can see there are unwanted objects are there. So we want only the selected object based on the shape. Based on the intensity, you can classify that. So once you did this, then you can go with any kind of machine language measurements. You can apply based on the diameter, or you can based on the intensity, or based on the size, and you can teach to the machine learning and try to elucidate your data. This is one of the example you can see. This is one of the mouse. I think it's a mouse testis actually. It's a cross section of it. You can see that they are trying to select it. This is called picture classification. So they are classifying. There are different types of nuclear sizes are here. So once it's classified as, then they told that these are the background. So we are teaching to the system to do the AI basically. So now we clicked it. Once after you selected this region, so these are the one immediately it's selected. You can see within a second it will selected which is the background, which is the non-background things, and it will be segmented properly. Now you can see the results, how it looks like. So now you can see, you can segment it properly. This is your 3-dimensional. This is your nucleus only. So you found only nucleus. There are unwanted debris or tissue or that everything is removed. In the 3-dimensional space, you found, see, this is the raw data and this is the segmented data. So once you segment this data, now you can see here. So sorry. one of the example I can show here. We were working with the yeast cells, and we are trying to study the endoplasmic reticulum circularity, because the scientist was applying different types of gene knockouts or gene alterations or gene deletion. They are trying to find how it is affecting the ER circularity. I don't know for what reason they do that, but they are trying to do. See, this is one of the yeast image. You can see this image. This image is very complicated. It's one of the very complicated image. You can see there are n number of yeast molecules are there. Everything is circular and it is co-localized with the nucleus, and they want to find this circularity. You can see this kind of circularity. They want to find this kind of circularity, everything. So now I started analyzing this data. You can see this. So once if I analyze this data, then I found the circularity and count and bounding height, width and everything. I'm trying to compare it with that actually, with outer ring and the inner ring. There are two different types of ring. We found it inside. One is inside, another one is outside. So we were very carefully segmented it. So this segmentation will take time usually. Means take in the sense like how you want to teach it. So these kind of rings I want, this I don't want. So like that you have to teach to the machine. Once you taught, then it is fine. You can go for batch analysis. That's absolutely no doubt. You can analyze like inner and outer very well. So once I analyze, then I can only plotted area. I have n number of parameters to plot it and compare with that. Is it the inner circle is small for this or outer? So now I can go with the inner circle and outer circle circularity, and I plotted into the graphical presentation, and I can see that how the variation in the data. Of course, there are one or two data will be outside that everyone know with, because that's the Gaussian usually. But you can see the Gaussian is totally different in both the cases of inner and the outer circle for one sample. Now you can see this is the another sample. For this second sample, we are trying to analyze again. Now you can see the inner and outer for the another sample with the gene knockout is totally different. But visualization, once you saw it, it's going to be the same. But once you go and analyze the data, it's totally different basically. So now you can see the circularity in both the cases, in the outer and the inner rings are totally different. And I am trying to plot everything together. So this is the another data for the B circularity analysis for the B sample. Now you can see I am trying to put A inner, A outer, and B inner, B outer. So in both the samples, there is a very huge variation is there. If you do the ANOVA for this, of course, you will see the huge difference. Just I'm showing you, this is one of the very minimal example, very minimal analysis. But this analysis takes me like initially for the segmentation, it took for me a little difficult. It takes maybe 1 or 2 hours to segment it. But once I segment, I apply for batches. Then afterwards, it's absolutely no problem. I can analyze very extensively. That is what one of the live example I want to share you. One more example I want to show you here in this in the live example. 1 minute. Can you see the screen? Is it the screen is visualized now? Is it the screen is visualized, sir? Now this is another screen. Is it visualized? No, no.
I can see the screen, but this is the PowerPoint screen.
Black color. Oh, still now the PowerPoint screen. 1 minute, sir. So now I want to show the screen. Yes. Now you can see the screen. Can you see the screen now? This is the IBS screen basically.
Yeah.
Okay. So this is a very interesting example. This image I took very recently, like 2 days before. In the neuroblastoma cell lines of the mouse, they were trying to study the mitochondrial dynamics basically. You can see these are the mitochondria in a very high resolution in the mitochondria. So you can see this mitochondria, how, and in the 3-dimensional space, I'm just showing you this mitochondria. So this is the 3-dimensional mitochondria. Okay. So in the whole cells, if you can go through, there are different mitochondria are there. And with a very high resolution, we can measure this mitochondria and measure the size. Now the question is, I want to analyze this mitochondria data to quantify that what is the size and how the volume, how the circularity. Is it possible? Means it is impossible for the regular any kind of data analysis we want to do, because it's a very huge data. You can see that it's a very big cell in the 3-dimensional space. We can do in timing also the same. This is a super resolution data. Now you can see here, I started analyzing this by using the IBS software. Now I can show you how this can be analyzed. You can see here. So we are segmented this. We are segmented this mitochondria nicely. You can see here. We segmented the whole mitochondria. So the mitochondria looks like this. So we were trying to segment the whole mitochondria. You can see here, the mitochondria is segmented here. So this segmentation is not easy basically. This is a very, very difficult segmentation. So, but we use this segmentation. Now the question is, okay, you segmented this mitochondria. Now the question is, what do you want to prove from this? What you are going to explain from this? So now you can see here, I went to the explanation here. So now the circularity of all this. So we found in this, there are more than around 2,665 mitochondria in one space actually. Okay. So what is the circularity of that? How the circularity is distributed in that? No. Okay. What about the area? If I would that, what about the volume? What about the count? If you have the mean intensity based on this, how the mean intensity, how the expression is distributed, you can plot everything within a second. That is the beauty of A-based analysis. You can go for the scatter plot. You can go for a different kind of plots here. You can see here, only with the surface area, you can plot it. And you can, and all this plot, everything, it's not going to do any other software. In the same software, you can analyze it. So, and you can analyze, you can get all these components. You can see surface area, volume, mean intensity, and you can go for the total intensity or the expression. Any details you want, you can get here in a very easy way. This is one of the example you can see here. You can go for the sphericity, you can go for the area, surface area, like any kind of component. So bound width or bound depth or bound height you want, like anything you want. Yes, you can analyze this here, and within you will get within a set. So now you can see all the data will be added here, like bound width, everything can be added here in this. So that's the speciality of the A-based analysis. Now you can see there is another plot I can show you. Now we can get the Pearson plot also. So with the mean intensity, yes, we can, and you can change this legend also. Actually, you can try to compare it how this is. So these are the very deeper analysis what the cell biology needed nowadays to conclude. So now people are trying to work with the oncology or different types of patient samples and compare with the control and the tissue, for example. See, if you have the oncology sample in the tongue oncology sample I use for the mouth cancer. So people cut the tongue from the good area as well as the tumor caused areas too. And they image it, they store it, and they give the images to me. So once I started analyzing these images, we found the new information. What are the variation between the control and the tumor area is there? And is it there common with the same population or different population, different sample? So that will be very, very interesting for us. So continuously we are doing by using IVI. This is one of the example I show you. This is the different types of violin plots and everything you can do for the comparison with the tissue or anything. You can analyze it. So these are the analysis we are doing continuously. This is one of the example I want to show you. That's I did. So now I can come to this the last part of the talk. I'm sorry. This is my presentation. Yes. So now you can see here. This is one of the example. You can see here. This is a regular HNS chain. Maybe you have seen these images a lot already. You can see that we are selecting here. So we are selecting this here. We are selecting here the area what we want to analyze. We are segmenting, because segmentation in the tissue is very difficult. You know that. So we have a lot of backgrounds. We have the area. So we are trying to segment it. So this segment happened within a second. Like once you feed the system, this is my background, this is my thing. Yes, immediately it will be set, and it's a very precise segmentation. It's not like that it can miss one or two. You can scrutinize that. If you have more background, still you have some background. Yes, you can see here. Now still you can cross, and you can do that and segment this actually. So now you can see once they do this segmentation, they can go for the analysis. Now this is an example. You can see with the different types of nucleus, they are trying to segment it. So this is A-based actually. So now they selected, and they told that these are the nucleus we want to study. You can see they selected this nucleus, and once they selected this, they feed it. See, this is the machine language they were selecting, and they can feed it actually. But this segmentation is based on you, like what you want to segment, what you want to analyze. That is very important. So it's not like that the machine know that you have to feed to the machine. That's very interesting. Any kind of images, you can give your pet images or any kind of images. So you want to segment and analyze based on that. Yes, we can do that. So that's the curiosity of the things. So now you can see immediately you can see the all once they click it. So the AI will be applied throughout this to the machine learning approach. Then immediately you can see here. Yes. Now you can see. Now you got all the nucleus were selected within a second. So you can, based on the diameter, based on the area, you can segment. That's also, there are n number of parameters are there to analyze and to segment also. Yes, you can do with area and this thing, you can segment it. So now you can see they are segmenting specifically based on the diameter. Yes, they were covered it, and now they can go for other one. So this is the, now you can see there are two different layers. They have two different proteins they can localize, and with a different color in the fluorescent microscope, then they can segment it properly. Like one by one, they are trying to segment it. And now you can see there is a circularity they want to measure. Yes, they can measure the circularity here. And they want odd, they want to it's remove the odd expression thing. Yes, you can, they can remove. Any of the dead cells you want to differentiate and remove, yes, you can. So this is the way. See, now it is easily identified where higher expression cells are there, where is lower expressions are there based on the intensity, based on the morphology. Yes, you can do. And you can go this here with the shape analysis. You can go the object recognition and trying to do the shapes. There are different types of shapes as there, different types of sizes. You can segment it separately and you can analyze why this kind of shape alterations there with this patient and other patients, or any parameters are changed. That's a reason. So the like that the analysis is going deeper and deeper to understand it. It's not like to give the treatment directly. Like now, this kind of analysis we are extensively doing here. Especially in my previous lab, I was working with National Cancer Tissue Biobank. We worked with the Apollo Cancer Center here and with different oncologists in the area. So we were, I was trying to analyze for them regularly. So we found there are very interesting information, and we are trying to compare this information based on our NGS data. Because you know that in NGS you come to know which protein, which gene is upregulated or downregulated. Then we can understand why these morphology alterations are there, why structurally these arrangements are there because of the gene, like the gene upgradation. So that is the way the analysis are going in deeper mode. So as at the last, so this is the picture classification process what we saw, and you can extract the information from any kind of fluorescence image and everything, and you can go for any kind of picture classification. Not only with this, but I used some kind of eye samples also with the tight junction proteins from Shankar Nethralaya and Agarwal and different R&Ds. We got the images and I analyzed that also as well. And these are the histology sample segmentation, because histology segmentation is very complicated. You know, this image, you want to segment anything, you want to understand anything is very difficult. Actually, these images are okay because these are our grown cells, but from the tissue segmentation is very difficult. Still, we can analyze this in a very detailed manner. This is one of the three-dimensional image of the super resolution of the cells. Actually, just I want to show you as an example. We this kind of organoid also we are trying to analyze. These are very big data. Still, we analyze this kind of organoid images in a very detailed manner. So, and this is the way we were doing it. These are some of the examples I just shown to you. And this is the electron microscope analysis. So for we can create and we can train the deep learning models. After that, we apply the models, and finally we can explore the results. So these are the different steps what we can do for the deep learning pipeline for the microscopy. I think my time is going to be over. I'll finish it soon. So this is the final stage of applying the different types of deep learning models are available with us. By using the deep learning models to understand different types of tissues, different types of cells, or different types of disease-oriented things. And I know that this is totally different for you, and it is new. But again, I'm sorry. I'm sorry, it is hanging again. Sorry. Can you hear me, sir?
Yes.
Is it that?
Yes.
I'm sorry, it was hanged. Don't mistake me. It's the final stage.
No, that's okay.
Sorry. No. Sorry, my PowerPoint was closed. Put the weightage. Okay, I can close this data. I'm sorry. Why this? Sorry, this is the last slide. I'll finish it. So I'm sorry, sir. Fine.
Dr. Raja, are you trying to share a slide?
Yes, yes, I'm exactly. I'm sorry because it was hanged, so
No
that's
problem.
why
Yeah.
it's not. Don't mistake me. 1 minute. This is the final thing, but suddenly it was hanged. It's not opening. I really don't know why. Zoom things was not opening for me. Zoom things are here, but is it there? I'm sorry. You are available?
Yes, yes, we are here. Yeah.
Right. Zoom workspace. I'm in the workspace only, but in the meeting, I'm not able to see anyone
Oh,
in
really?
Zoom meeting. No, because I'm sorry. I think something happened, but your things are there. Finally I got you. Finally I got you. Is it fine now?
Yes, it's coming. Yes, we see it.
I'm sorry. I'm sorry. We are in the last. Because of a lot of videos, I think that's the reason it is. I'm in the last slide only. I'm in the last slide. Just I want to finish it here. Yes. So as I told you, there are different types of segmentation are there. So different cells, you can see this is the cell imaging library. We are segmenting in a very, very detailed one, because this kind of segmentations, it's very difficult. And our customers and the scientists are really happy once we do this kind of segmentation. So this kind of cell post based segmentation we are doing continuously in membrane level or the protein level or any kind of DNA structure, cytoplasm, nuclei. So everything can be segmented properly. So that's the very interesting. And we have the deep learning things, and we call this as autonomous microscopy. Means once you take the image, immediately you can give the IBI data to give the results also directly. So it's like autonomous. So it's a very new one in the world we introduced to take the image and analyze and show you by using AI in a very, very drastic manner. It's not like that you are sit and analyze. You can feed it and analyze directly. So that's what we are trying to do it here. And this is the workflow for creating this kind of analysis and take the data and analyze it immediately. Okay, this is the autonomous one. So if you found the data and any kind of rare events, it will automatically check by using AI and analyze and show you. So that's the beauty of the system. Okay. So now this is one of the last example of endocytotic vesicles. We saw that you can see that endocytotic vesicle can be moved, and due to the time it's changing because the pH is altering, so the color is changing. So by using the film microscopy, so now we are capable to see the different types of the time-based one alteration, and based on the pH alterations, we found the movements are moving, like changing actually. So we can try to find in a very molecular level inside how this is moving and how we are controlling this, so we can analyze that. So this is one of the best example for doing the very deeper advanced cell biology applications. Just we want to show you like these are the deep learning approaches we are doing by using IVR. Thank you so much for the attention. I'm sorry if in between it was hanged. Thank you for it. Please, you have any questions?
Sure. So thank you so much, Dr. Raja. This was very excellent talk,
Thank
images
you.
and software demonstration. I loved it personally. Very impressive and
Thank you.
shows a great potential, I think, to advance, precision medicine as we were talking before the talk. Super
Yes.
Science microscopy, which is your specialization, I think, along with the stitching, those are the two concepts.
Yes.
I really liked it, and it was very easy for
Thank you.
someone who is not too much into biology to understand the
Correct.
images techniques. So we
Correct.
appreciate your sharing expertise. We would like
Thank
to
you.
open the floor for questions now. I see already couple questions in the chat, so
Oh,
let
really?
yeah.
Okay.
Yeah. So we'll start with Srikant Solo's question. Is
Yes.
Avia an image processing software? Can it be used for imaging data? Does does it come into picture post segmentation or even for segmentation?
Yes, like we are not able to capturing the images there, but we can analyzing the imaging data. Like you have any TIFF images or any JPEG images or anything you have, any the raw data you have, we can try to analyze that.
Okay.
So you can do any kind of segmentation. Absolutely fine, no problem.
Mhm. Then a question from Dr. Raghuveer Halkar.
Yes,
Great talk and images. As physicians,
thank
we
you.
are interested in increasing the specificity of the medicine we use or investigations we use, such that side effect is minimal. Targeting at
Correct,
molecular level was envisioned thinking that it will be more specific than targeting at organ level or even a cellular level.
correct,
We are
correct,
finding even at molecular level, the nature is not very specific. Good example is
correct,
PSA-based theranostics. Is
correct.
going more at microscopic or deeper and deeper level helpful? Will it increase specificity or it will be zero and one like in computers? Is holistic approach better? Is the big question.
Wow, this is a great. It's a great question. I have to thank you because practically, of course, he's a clinician. So here in India, I was working with the great oncologists, some of the people from Apollo, and we have a very long discussion. The same discussion. Sebastian, we are giving medicine, so we treated the medicine based on the basic results. What are the tests we saw? Then we tried because of the age, the dose concentration will change. But now the question is, so the drug what you are giving to the X person can be like studied by using at the molecular level only. So once it is molecular level, it pass, then we will go for the clinical level that with like any kind of like clinical research, CROs and everything people are doing, and finally it come to the market. But all the data, what we were found to understand into the molecular level, now it is the game point. For example, NGS, whenever it started, it is too costly, anyone can do. But now in India, we have, like, from my knowledge, we have more than a lakh of NGS machines. Because India is highly populated, more disease, patient level is high. Almost most of the people, they now everyone is doing the NGS. Once you do the NGS, of course, it is a molecular level. So recently, we posted from our IIT Madras, Dr. Malingam, I worked. We posted a National Cancer Tissue Biobank as a breast cancer NGS data for Asian population. So they studied more than like 1,000 people, because doing the 1,000 samples for NGS is not easy. It's very difficult. The data is very huge. It's in TBs and TVs. You have to analyze the whole genome sequence of every individual and trying to compare with that, and it's a very huge data. And now they are giving the drugs based on that, because they call nowadays they are treating the cancer is a genetic disorder, like that they are coming. So these are the genes, or these genes are upregulated or downregulated in specific age time, or any food practice or anything, it's induced. Of course, you will get to the cancer at this age, like that they are predicting. It's not like I'm not telling it one patient or anything. So nowadays, all the molecular level analysis have started coming. So now the people are turning to that. Of course, beyond all this thing, as sir says, the holistic approach is there. in medicine. We have to accept that is beyond the human control, so we understand, but another question also there. Thank you for it. Is it by using this software will have the notes in pet image? Yes, you can. With the pet images, I have to try. I got CT images from some people in National Institute of Immunology. I tried that, and with their raw data is not supporting. But if you have any kind of two-dimensional data, or otherwise you have any data, please share with me. I can try for it. I'm interested to try for it. We can try and see. Of course, any kind of images, because we have very strong segmentation. Okay. So if it is a pet image, we do the threshold analysis, and then we can absolutely we can do that. I saw Dr. Maitri Shankar as raise hand, sir. Maybe anything.
Yeah, hi Raja, how are you? You've always
Hello.
had a
Thank you, ma'am.
you've always had a knack with images, and I still have the picture we did together on in my office, the lung cancer and the smoking
Okay.
art.
Okay.
Great talk, very colorful
Thank you.
images. My question
Yes.
is, when you say all this pet image analysis, this is like in the omics space, right? Radiomics and genomics, what we
Correct.
are talking about these days.
Correct.
Is this also similar to what we are looking at telomere length when we are looking comparing biological age to chronological age in a longevity perspective? This is also being done in the same technology, I'm presuming. Having said that, knowing that there is a lot of cognitive bias when we deal with AI. AI has a lot of cognitive bias, and when we are working in an area we know so little about, the feed is coming mainly from electronic microscope. How reliable is the information, or what is the clinical relevance? This is almost like doing biopsy in vivo, real time.
Correct.
Right.
Correct.
So how
Correct.
are you actually doing it? Are you it should be a live sample. So I'm trying to understand the clinical relevance of it in everyday practice. Yeah.
Correct, ma'am. So this is a very important question as you talk about the bias. Just I'm telling you, before concluding something, minimum we will see. For example, in cell biology, anything you want to conclude, you need minimum n number 100. Minimum, I'm telling you, 100. Okay. So it will be 100 cells, or otherwise, if it is a clinical sample, 100 patients minimum, and trying to find the bias from that, how the tissue or how the image is and imaging condition, everything should be constant. It's not like that you can randomly take some image, you can analyze. No, it's a very huge data, especially with the clinical thing. You want to conclude anything, we have to spend a very huge time on that. So once we understand with the control, and we know that we check with all the parameters. If you give the threshold level this much, what is the result? Threshold is low, what is the results? And try to compare which is the best one. So this we take help from clinicians as well. Once we analyze, we discuss with clinicians, and finally we conclude, okay, so this is the correct. Now, then we can go for other samples and we can try to conclude. So for the bias, yes, of course, for biology, bias is very high. But A means we are trying to take the help for the very large data analysis, not for smaller one. Okay. So in the large data, you can segment it. You don't want these kind of cells, you don't want. That is wrong cells. Means that information we are getting from clinician. Okay. So these are the correct scorer cells. So you can analyze this. So once you feed it this, then the system will be learn nicely, and you can go for a number of batches. Of course, bias is a very serious issue. We have to sit and do that.
Who are your customers right now? I mean, what kind of cases are you taking, or who is the service?
Very good. Yes. Now at this moment, because IVIA we introduced a year before in India, because I joined with Leica like 1.5 years before. Before that, they didn't take action because I'm the one who lifted. So we sold, one of the example I told you, we sold in a very advanced, very latest biopharma company. Okay. So what they're doing is they synthesized the molecules, oncology molecules, around like 1,000, more than 1,000 molecules per year, I'm telling you, by using chemist. They are checking only DNA repair. Understand? Only DNA repair. They bought the microscope from us. They got the images. Images are stable and everything in the cells they are checking. They are checking only the DNA repair amount, the quantification. For quantification, they use our software. DNA repair happened only in the nucleus, inside the nucleus only, it's not in anywhere. So inside the nucleus, they saw, then they can quantify that, how much is there, and they can go for batch and batches. So in different population, like different concentration of drug, they do. This is one of the example in biopharma. Okay. The similar way, now some of the clinicians, especially or the pathologists, who are doing the tissue imaging for brain or for cancer or anything, they're coming. So for them, then we are giving, yes, there also people are doing it. And the third case, some people are working for host pathogen interaction. One of the scientists from Center for DNA Fingerprinting, we know. So that guy is working on E. coli, is a virulence E. coli who is attacking the brain, So once after the infection, how the E. coli is infected the cells, it is creating new kind of wobbles inside, like some kind of vacuoles inside. So we are trying to quantify by using this software. So
Yeah,
these
and
are
how
the three various.
are these machines? How expensive are these machines?
Absolutely, this is expensive because it's a very detailed analysis. The start pack is cost around 30 lakhs.
Okay. And in the live, you're showing, right, the cells moving and all that. Like actually, the
Yes, ma'am.
is that like in animal models or in real life?
No. This is the grown cells. This is the grown cells. In animal model, that is the developmental biology.
Grown
That is
specimen.
different.
Okay. Understood.
That is whole specimen only. There you can target a protein. For example, the C. elegans, yes, you can. Drosophila, yes, you can. But for any other big animals, it's very difficult. Otherwise they will go for the organelle imaging, like PET, like this kind of CT tomography. People use that, and we have to analyze that. Yes, that's it.
Sure. Thank you.
Thank you, ma'am. Thank you. Dr. Amul. So the moment we grow up in all cells are one piece of having normal CT for 45-year-old person almost. They may not have the cancer, but have a lot of other weaning pathologies. So the TIL control for A hard to find how to around it. This is a very, very good question. Again, for the CT, I'm not. I never analyze much CT images, so that's why I can't immediately tell that these are the parameters you can avoid and you can find. But with A, yes, you can. But with CT image, you have anything? Please share with me. I can try to understand and discuss with you. Then we can because without seeing images, we can't tell anything. In the
Yeah,
case
but
of
even
CT,
even for PET, you would see the same thing, right? There is a lot of uptake because our tracer is not disease specific.
correct.
For example, FDG
Correct.
is showing glucose metabolism, not necessarily cancer. As Dr. Halkar pointed out, PSMA is not prostate specific. It lights up in a lot of other tumors and pathologies. So it's hard for us to get a real control for the AI black box to pick up what's normal and what's abnormal. And nobody knows how these AI systems are being trained. Like you do some inputs and then it comes back as an output, and we really don't understand what it is considering normal and abnormal. It's almost like voodoo black magic sometimes. So without knowing how it's making those decisions, it's hard to trust it, especially in the era where we are learning more. Like we are learning so much that we don't know a lot about medicine. The more we know, the more we don't, kind of a thing. So that's why I was curious how people are relying on AI for these things.
This is absolutely perfect because we were practiced with some kind of home protocol already. So the new one which is coming, of course, there are a lot of bottlenecks. We didn't understand where we are doing. Usually AI looks like it is like immediately you'll get the results. We can't believe that how it has happened. For me also, once in cell biology it's happened. As a clinician, it will be more serious because you are directly treated with the human beings. So if anything will go wrong, it's going to affect you. So that's why these kind of, but again, for the PET image analysis by using AI, we have to sit with the PET images. Once we sit with the PET images, we can elucidate it and we can find it. It's not very difficult compared to cell biology. I'm sure, because the whole organ imaging, it's a full image, but we have to see the images. And we have to apply with the AI algorithm and try to feed it and understand we are doing correct or not with the controls and with the expert of the clinicians like you. then we can control it. But nowadays it is moving towards to the direction. Anyway, in future it's going to be with that, because people want to do immediately, especially now with all the oral stuffs. Yesterday also I saw in the Savita dental college here, the people have the oral. Once after the operation, they have the whole jaw image, and they were trying to put it into the things. So they build the jaw actually based on the age. Also, the jaw size is changing, the tooth size is changing. So they have different types of AI-based algorithm they applied. Then they're moving nowadays. But especially as you say with the CT images, we have to see. I'm not exactly it's working on it still now, so I don't know. But do you have any images? Let me know. I can see that. We can try to understand.
Sounds
I'm
good.
sure the future it will be very useful for you. And you see, once upon a time, no X-ray. After that, every labs we have, every hospital we have X-rays. Similar way, now the CT came initially. Now the analysis of AI will integrate, and people will do very easy, because we know every day we are going very easy and easy. So AI is going to be like that.
Yeah.
It's not
I
like
would
that
be,
AI
yeah.
never beat us, frankly. AI never beat us. The only thing is what we are feeding to the machine is important.
Yeah.
So if you feed the correct thing, that will helpful for you to go very speed analysis. That's it.
Yeah. So I would be
Frankly
interested in,
speaking,
seeing if your
yes, sir.
system can analyze, say, for example, PET imaging data for amyloid imaging with pre
Yes,
and post anti-amyloid therapy, and see what
correct.
it comes up with. I would be very interested in seeing, because our
Um,
current quantitative measures, I think, are lacking in a lot of things. So
oh, really?
I'll
I'm so
connect
happy. If you
offline.
have the images, please share me. If you have the images, please share me. I can try, sir. No problem. I can try to see, because I'm also interested how to analyze these images in this. I would have been really happy. So
Sounds good.
if you have the images, please. You are from which place, sir?
Emory
You
University in
you are
Atlanta.
located? Okay, great. We'll see, sir. We'll see. Please share. We'll see that. And we have a team, sir, team there. We'll work.
Okay,
Thank you.
thank you everyone. Thank you, Dr. Raja, again for
Thank you,
fantastic.
Thank
Deepak.
you. Great talk.
Yeah.
Thank you so
Thank
much.
you.
Thank you so much for
Thank you,
everyone.
Dr. Raja. Yeah,
Thank you.
excellent talk.
Thank you, man. Thank you so much, Deepak. Thank you, all of them. It's
Thank
a
you.
big honor for me. See you again. Thank you. Thank you so
Okay,
much.
thank you.
Bye.
Thank